Hello, everyone. My name is Kit Rowe. I'm the Vice President of Sales and Marketing for Tiger-Sul Products. Tiger-Sul Products is a global leader in sulfur bentonite. We've built our reputation on quality products, customer service, and our commitment to research and education. From the entire Tiger-Sul team, thank you for joining us for today's webinar, Enhanced Crop Nutrient Utilization with Sulphur. With me today is our Senior Agronomist, Wesley Haun. Wesley coordinates research and development on soil fertility and crop nutrients in the U.S., Canada, and internationally. He conducts education programs that emphasize the benefits of complete plant nutrition programs. Wesley received his Bachelor of Science in Agricultural Mechanization and a Master's in Plant Soil Science from the University of Tennessee. He is a certified professional agronomist, certified professional soil scientist, and a certified crop advisor. He has a diverse agronomic background that spans retail sales and consulting at the farm gate, as well as agronomist at regional, national, and international levels. With that, I will hand it over to Wesley.
Thank you, Kit, and welcome to everyone who has joined us this afternoon as I present information on enhanced crop nutrient utilization. The whole idea of this presentation is to help you better understand the influence that sulfur may have on plant metabolism. Sulfur's role in the plant. Amino acids are the building blocks for proteins, and sulfur is a central component of two of those amino acids, cysteine and methionine. Sulfur also impacts protein quality in forages. Adding sulfur tends to increase protein. Sulfur is also involved in enzyme activity, as well as the photosynthetic process and the nitrogen fixation in legume plants. We as agronomists, and probably you as salespeople in the crop production, we strive to maximize crop yields, but oftentimes what is out of sight is out of mind. We tend to overlook those dynamic soil interactions, whether it's the physical, the chemical, or the biological, and also even the plant that's above ground and what's going on internally, all tend to influence the availability of nutrients for crop uptake. Various nutrient uptake mechanisms are involved, and nutrient availability at that soil-root interface is controlled by soil properties, root characteristics, such as is it a fibrous root system or is it a taproot system? The crop growth that's taking place at a given point in time in the growing season, as well as the nutrient chemical reactions that take place in the soil prior to uptake, and then the presence of soil microorganisms. All these factors influence that root to soil interface we call the rhizosphere. The rhizosphere is that first point of nutrient uptake. Interactions is at this interface, and the rhizosphere actually is a zone of very intense microbial activity, and it's critical to the soil and plant relationship. Roots release exudates, or in other terms, leak, energy-rich compounds of sugars, organic acids, and to which microbes are attracted and feed upon. This rhizosphere includes the root, as well as the little projections you see around that root we call root hairs. So as you can see on the right side of the slide, the root hairs on a nodal root of a V2 corn seedling, and this is a very small corn seedling, and notice the intensity of those root hairs. These root hairs enhance the ability of the roots to take up water as well as various nutrients. And then we have the functional bacteria in the soil, and those in the nitrogen fixing bacteria in legume plants such as your soybeans, alfalfa, clover, and the peas. Legume nitrogen fixation nodules, and what we have is rhizobia bacteria in the soil infect the roots of legume plants and cause an infection that actually creates a little structures we call nodules on the roots themselves. As you can see in this particular slide, the nodules on an alfalfa plant, and these are nodules on a soybean plant. And if you take a nodule and increase the size of that such you can actually see it and slice one of those nodules open, if it has a pinkish red color, that's an indication that the Rhizosphere bacteria is converting atmospheric nitrogen to ammonia, which then is converted to ammonium that the plants can utilize. This relationship is actually called a symbiotic, which means that the Rhizosphere bacteria is providing nitrogen for the plant, and the plant in turn provides carbohydrates for the Rhizosphere bacteria to live and survive. These nodules only live for about, or active for about 30 days, and they slough off, and new nodules will form in their place and repeat the whole process. So this goes on throughout the growing season. But I might note within those nodules on a legume plant, one of the amino acids, cysteine, is actually starting to create in that nodule and then translocate into the plant for further use in building proteins. So crop uptake. These are the nutrients that crops and plants require to grow and to maximize its yield potential. Whereas the term crop removal refers to those nutrients that leave the field at harvest time. Crop uptake rates are somewhat higher than crop removal rates, and sulfur application based on removal rates can result in a potential yield reduction. In this table, we see some selected crops and the crop sulfate requirements for each. And for example, the corn at 0.18 pounds of sulfate required per bushel, and with a yield of 175 bushel, we have an uptake demand of 32 pounds of sulfate per acre, whereas at harvest time we're only removing 14 pounds per acre. Move down to the cotton, and notice for a challenging 3 bales per acre yield, requiring 15 pounds of sulfate per bale, provides an uptake of 45 pounds per acre of sulfate, whereas at harvest, only 8 pounds of sulfate is being removed from the field. Point we need to make here is, if we make a recommendation for sulfate for a crop based upon removal, are we going to have enough that our supply of that crop throughout the growing season, especially during its peak demand? So with that, I suggest we take a look at recommending sulfate requirements based upon potential uptake needs. And that plant uptake is regulated by plant demand. It's facilitated by those root extrudates, the sugars, the proteins, and the organic acids that are leaked from the plant roots. They also serve as nutrient carriers across that root cell walls. And, as mentioned earlier, provides a food source for the microbes within that rhizosphere. By evaluating soil sulfur availability to the crop, it's more challenging than other nutrients, mainly because we have multiple sources providing sulfur to that soil ecosystem. We have that carryover potential from previous crops. We have plants that can store sulfur internally but cannot translocate it within the plant. And then sulfur translocation with that plant is minimal. And then we have various loss mechanisms that compound the situation of trying to estimate sulfur demands and needs. Those include leaching, volatilization, surface loss and erosion, or with erosion, immobilization, mineralization, which results in some insoluble forms, as well as crop removal. And plant nutrient efficiency. Here again, trying to balance the sulfur for that whole plant. The demand function is based upon developmental growth stage, whether it's in the vegetative growth or in the reproductive growth stage. There again, sulfate uptake, the distribution, and assimilation balanced by the plant's demand for growth. There are various means in which plant nutrient uptake occurs. The three main ones are mass flow, diffusion, and root interception. Mass flow is where solutes, that is, nutrients that are dissolved in soil water, called solutes. Once that soil water moves across a root mass, it is available for uptake by the root mass. Then we have diffusion. This is the movement of nutrients across that root cell wall, going from an area of high or low concentration into another area of opposite concentration levels. And then root interception is as the roots grow through the soil and incidentally tap into a nutrient, some uptake can take place. But the two primary means of uptake is mass flow and diffusion, as illustrated in this table, with the primary and the secondary nutrients. And notice with nitrogen, mass flow is about 80% of the uptake occurs as compared to 19% with diffusion. Whereas with phosphorus, 93% is diffusion versus about 5% with mass flow. And same for potassium. It's about 80% diffusion, 18% mass flow. Whereas with sulfate, it is 98% through mass flow. Here again, sulfate is very soluble in the soil and flows with soil water. And then you have calcium and magnesium, both predominantly mass flow as well. Interesting part about the calcium is 40% can be through root interception, whereas though the other nutrients, root interception is very minor in its uptake methods. And ion uptake. Here ions can be considered cations, which are positive, or anions, which are negative. When we say ions, we're talking about either of the two. Nutrient uptake is selective. Here again, plants do not take up all nutrients at the same time. It's based upon the crop demand at a given point in time. The uptake of some ions must cross a concentration gradient, and the uptake requires energy for some of those nutrients, which is supported by photosynthesis. The uptake is a function of transpiration, which actually serves as a nutrient pump. From this point forward, let's assess how plants actually take nutrients into the root and move it into the rest of the plant. In this slide, we have a cross-section of a plant root, and notice at the bottom is the root cap, and just above that root cap is a condensed layer of cells. This is the apical meristem region, which is actually the growing point, and just above that is the elongation section. This is where the cells actually grow and expand and mature. And just above that are the mature region of the root, and it's this region where the root hairs actually develop and differentiate. Here we have a cross-section of the root. And notice the outside ring is the epidermal layer, which is the cell wall, and the projections would be the root hairs. Living tissue, including plants, is negatively charged. But notice energy from photosynthesis with ADP interacts with the hydrogen, which is positively charged within that root, and forces it through the root cell and outside of the root itself. Here we have a net negative charge, and then the cations in the region around that root will be attracted and move across that cell membrane into the root itself. But note that the hydrogen that was actually removed from the interior of the root, here again, will connect with the sulfate, which is negatively charged, and serve as a nutrient carrier and help move the sulfate across the cell membrane into the root itself. Nutrient movement. One method is called passive transport, and it is diffusion movement of ions across a membrane. In this situation, it is the root cell wall from side of higher concentration of ions to side of lower concentration of ions. This is called moving with the concentration. Only a few molecules can actually pass directly through most cell layers of leaves as well as the root cell walls, such as oxygen or carbon dioxide. Glucose and water need some assistance, referred to as facilitated diffusion, as illustrated on the right side of the slide. Protein molecules, which is the yellowish-orange section you see there around the arrow, forms a bridge or a passageway through that root cell wall that permits easy movement of water as well as some nutrients, as illustrated in the previous slide with the sulfate moving with the hydrogen. Another method of plant nutrient movement is called active transport. Active transport of nutrients occurs across the membrane against a concentration gradient. That is, from an area of low concentration to an area of high concentration. This requires energy to move the ions into the cell. This process uses ATP energy acquired from respiration, which I will explain later. Protein molecules form a bridge through the root cell wall, which acts as a catch and release mechanism. Nutrient ions move from the low concentration region and enter into the bridge. ATP energy then closes the bridge on the receiving end and then opens on the deposit side to release the ions into the cell. Then we have nutrient carriers. As I mentioned earlier, you have the proteins with amino acids, you have organic acids and sugars as well that can facilitate movement of nutrients. Sulfate is a component of protein carriers, both in higher level plants as well as lower level plants. Photosynthesis process is complex. It utilizes water, carbon dioxide with solar energy as sunlight to form glucose and oxygen. This process absorbs the energy as sunlight. Photosynthesis sustains life, not only for plants, but also as mammals. It occurs within the chlorophyll pigments contained within the chloroplast. Here we have the leaf structure, and notice at the top the cuticle is a waxy layer, and then just under that is a single layer of epidermal cells, which is referred to as the cell wall. And just under that, you have the mesophyll cells, and this is where the chloroplast, which contains the chlorophyll, is located. Photosynthesis takes place. Sulfur does influence photosynthesis. In a research project by Terry at the University of California in Berkeley back in 1976, demonstrated that in the absence of sulfur, we had a reduced amount of photosynthesis due to less chlorophyll being present and a decrease in the carbon dioxide uptake. And I may note at this point, plants and animals work together. Plants utilize carbon dioxide just like you and I utilize and breathe oxygen. Plants take in carbon dioxide and release oxygen, whereas we as mammals and humans take in oxygen and give off carbon dioxide. Sulfur does influence photosynthesis. This is a trial with rice, demonstrating where chlorophyll absorbs the sunlight, and if sulfur is deficient, there was a reduction of 49% in the amount of chlorophyll content. This project also demonstrated that the functionality of photosystem 1 and photosystem 2 within the photosynthetic process were reduced. System 1 had a reduction of 61% in efficiency. System 2 had a reduction of 31%. Let me expound on this. Photosystem 1 is considered the light reaction, whereas photosystem 2 is considered the dark reaction in the photosynthetic process. And hence, that's the reason we see a greater reduction in the efficiency of System 1 with the reduced amount of sulfur being present. In plant respiration, which is essentially the opposite of photosynthesis. Respiration utilizes the glucose that was produced in the photosynthetic process with oxygen, produces carbon dioxide, water, and thermal energy as ATP. Generation of ATP is often referred to as photophosphorylation. It's interesting to note that the plant only needs about 20% of that energy that's created. Therefore, the other 80% is applied to dry matter accumulation, which ultimately leads to food and fiber production. So sulfate interacts with ATP to form various enzymes, esters, and other sulfur compounds utilized in the plant's metabolic processes. Plant transpiration serves as a pumping action to move plant sap from the roots up to the leaves. The extent of this activity is a function of ambient temperature, relative humidity, wind, air movement, amount of soil moisture, and of course the type of plants. Plants will transpire more water than its own weight, oftentimes in significantly greater amounts. For example, 1 acre of corn can transpire approximately 3,000 to 4,000 gallons of water per day. 1 large oak tree may transpire more than 40,000 gallons of water per year. The process of water movement from the soil through the plant and loss from the leaf surface by evaporation is called transpiration. This drives the long-distance movement of water and nutrients throughout the plant. Sulfur does interact with other nutrients as well. Specifically, nitrogen and sulfur are synergistic. Both are involved in the protein synthesis process. Sulfur regulates nitrogen metabolism. The lack of sulfur results in reduced nitrogen use efficiency. The maximum benefit of nitrogen requires sufficient sulfur to supply throughout the growing season. So a lack of nitrogen results in reduced sulfur use efficiency. Sulfur has a synergistic effect on crop phosphorus response as well, which is a function of the soil fertility status, the amount of sulfur that was added, the specific crop being grown, as well as the weather during the growing season. Another study was conducted looking at the interactive effects of nitrogen, phosphorus, potassium, sulfur, boron, and zinc in rice, where an omission trial was established. And what I mean by an omission trial is you set up the trial with all the nutrients and then start removing one at a time to evaluate a specific effect by that nutrient. In this case, 25% yield reduction occurred when sulfur was removed, or a 2% reduction in yield occurred when boron was removed, and a 14% reduction in yield occurred when zinc was removed. So evaluating some trials that Tiger has conducted or is in the process of conducting, this trial is over 2 years on potatoes, and note that the sulfur from Tiger 90 and the Tiger XP yielded 37 and 32 100-pound units, greater than the grower standard, which is represented by the GST. But I might note that the Tiger magnesium yielded 58 100-pound units more per acre. So that shows when sulfur and magnesium jointly revealed a much greater yield response than sulfur by itself. This is an onion trial that's also in the process and has not been terminated. Here again, this was conducted in a grower's commercial production field using his normal production practices, excluding sulfur and the micronutrients in question. In this case, we saw a slight increase when zinc was added, another increase with manganese, but a much greater increase when the onion mix was added, which contained both the manganese and the zinc. So this shows when you add the sulfur along with other nutrients, we saw a much greater response. So in summary, sulfur is a critical component of many plant metabolic processes. Sulfur facilitates the uptake of various nutrients by plant roots. Crop sulfur needs to be based upon crop nutrient uptake rather than removal. Nitrogen and sulfur are interactive, and sulfur can influence crop response of other nutrients. So hopefully this provides some insight as how sulfur can enhance extra crop growth and nutrient uptake. If you need further information, I encourage you to contact your local account manager, or you may contact me directly through the email shown on the slide. So with that, Kit, I will turn the program back to you.
Thank you, Wes. As a reminder, if you do have a question, click on the Ask a Question button in the upper right-hand corner of your screen. Here's our first question. Wes, why are you recommending higher sulfur application rates than some research reports are suggesting?
Kit, I can appreciate that concern, and I prefer to make a sulfur recommendation based upon crop uptake requirements. There's various literature published indicating what the crop demand is for various crops on a sulfur basis. And then there's other publications that have crop removal rates. And with some crops, that's a pretty significant difference between what the crop actually is removing nutrients from the field versus what that total crop needs are. So I prefer to be conservative and apply what the potential crop uptake requirement is going to be rather than depending upon the amount that we actually remove from the field. And this is very similar to nitrogen recommendations that we normally follow as well.
Thanks, Wes. Here's a second question. I am not sure I understand the direct role sulfur has towards nutrient uptake. Can you explain further?
I will try to expound further there, Kit. And actually, it's not a direct role as much as it is an indirect role. Remember, as I was talking about how the ions are moved across the root cell membranes with facilitated assistance such as the diffusion process or going across a concentration gradient. There are proteins that build bridges across that cell wall. Proteins, remember, contain amino acids, and sulfur is a component of two of those amino acids. So here again, in a more indirect role, it is supplementing the assistance of moving ions across that cell wall.
Thank you, Wes. Here's another question. Looks like it's coming from somewhere in the southeast. How does transpiration move water and nutrients up the plant?
Interesting. You know, mammals and humans have hearts to circulate their blood within our bodies. Plants do not have that. Whereas with the transpiration process, moisture evaporates off the leaf surface, which creates more of a vacuum or serves as a pump to draw moisture from the roots up through the plant and then lost through evaporation from the leaves.
Very good. Thanks, Wes. Here's another one. Can you explain further how sulfur helps a plant take up other nutrients?
The specific mechanisms of many nutrient reactions with sulfur are not fully understood. A number of published research documents, as well as our own Tiger-Sul research, has demonstrated crop yield responses with sulfur as well as other nutrients. Now, some of these responses tend to be crop specific, but the more interactive responses that we have observed tend to be sulfur when combined with nitrogen, phosphorus, zinc, manganese, and magnesium.
Okay, Wes, looks like we've got time for one more question. Why is root interception so low for nutrient uptake?
That's a good question. Root interception is a direct nutrient contact by that root as it explores the soil profile. The total amount of nutrient contact by the root is actually very small compared to the total soil volume. And root nutrient uptake does not occur through the root cap as that root continues to grow.
Thank you, Wes. In closing, I want to thank all of you for joining us today. A recorded version of this webinar will be available on our website near the end of the week. Anyone who watches and needs CCA, CEU credits can self-report. Anytime you have additional questions, you can always visit our website. Once there, you can find your local account representative or simply click on the Contact Us link. If you include your name and an email address, we will respond promptly. Also, check out our website for additional information on sulfur bentonite products, including our line of micronutrients. You can also request access to Tiger University, where you can see white papers, newsletters, and other technical bulletins. Thanks again for joining us today. Have a great week.